Assalamu student. My today's topic is development of chick. Why do we study development of chick? The pattern of development of chick is similar to the pattern of the development of various vertebrate eggs. That's why we study the developmental pattern of the chick. In the previous lecture, I have explained the structure of a hen's egg. Hen's egg is polyvesical or is also termed as megalocecal and microcecal. As well as it is meolecycal, means it consists of large amount of yolk that spread throughout the egg, in the result of which only small portion leave, which is termed as blastodisc. This blastodisc is also termed as germinal disc or active cytoplasm. The egg of hen is formed by the process of oogenesis in the female reproductive organ, which is termed as ovary. The egg released from the ovary in the form of secondary oocyte. This secondary oocyte, when released in the oviduct, then it is further passes to the mitotic cell division, in the result of which two polar bodies is formed, which is later on degenerated. Before the secondary mitotic division, fertilization is take place in the oviduct upper part, where two cells are united together, that is male and female sex cells united with each other, in the result of which single diploid cell is formed, which is termed as zygote. When zygote is formed, then the process of secondary mitotic division take place and the polar bodies are formed. These two polar bodies later on degenerated and mature ovum as passes through the oviduct. This mature ovum is later on surrounded by the proteinous material, which is termed as albumin. This albumin surrounds the egg outside the vitelline membrane. Here the vitelline membrane is present around the egg, and outside of the egg, albumin is surround egg. Outside the albumin, two shell membrane surround the albumin, and after it, another calcareous, porous, hard material surround the egg to protect it from the surroundings. The fertilization in the hen is internal, and the egg is laid after the 24 hours of fertilization. When the fertilization takes place within the egg, then the second event is started within the egg of hen, which is termed as cleavage. Here are different patterns or events that are take place in the development of chick. One is the fertilization, then cleavage, then morula, then blastula, then gastrulation, and then neuralization. When the fertilization takes place in the egg or ovum of the hen, then second event has been started, which is termed as cleavage. What is cleavage? Cleavage is the repeated mitotic division that is take place in the blastodist, in the result of which cells are formed which is termed as blastomeres. There are two types of cleavage that take place in the living organisms. One is holoblastic, another one is the meroblastic. Holoblastic. Holoblastic is also termed as complete cleavage because the egg of the animal is divided completely. Means the whole egg is cleaved completely. That's why we termed it as a holoblastic cleavage. The examples of holoblastic cleavage are isolacetal, which is also termed as homolacetal, and mesolacetal. These two types of eggs are considered the example of holoblastic cleavage. While meroblastic cleavage is the cleavage in which only small portion of the egg undergo the stage of cleavage. That's why it is also termed as incomplete cleavage. The examples of meroblastic cleavage are telolacetal and centrolacetal types of egg. Meroblastic cleavage is further divided into three types. One is the bilateral cleavage, second is the discoidal cleavage, and third is the superficial cleavage. Bilateral cleavage. Bilateral cleavage is the cleavage in which cleavage takes place bilaterally, and the example is cephalopods egg. While discoidal cleavage takes place in the fishes, birds and reptiles, in which cleavage takes place in the disc of egg. That is blastodisc or germinal disc. While third cleavage is superficial cleavage takes place in the insect, in which cleavage takes place on the surface of the egg. Only on the surface of the egg cleavage takes place, while the remaining portion of the egg remains as it is. So, in hen, the cleavage that takes place is meroblastic discoidal cleavage, in which cleavage takes place only in the blastodisc, while the remaining portion of the egg remains as it is. Only the cleavage takes place in this blastodisc. Here is the above view of the blastodisc, in which you can see that the first cleavage is on the vertical plane and the second cleavage is also on the vertical plane, but it is on the right angle of the first cleavage. Means when first cleavage is formed, on the right angle second cleavage is formed. And the result of this, 4 cells are formed, which is termed as blastomere. The third cleavage is horizontally take place. The first two cleavages take place vertically, means like this, vertically, while third cleavage is take place horizontally. In the result of first two cleavages, 4 blastomeres were formed, while because of horizontal cleavage, 8 blastomeres are formed. 4 on the upper and 4 on the lower side of the blastodisc. While the remaining cleavage is irregular, in the result of which delimited cells are formed and are termed as blastoderm. Means blastoderm is formed in the result of irregular pattern of cleavage, and number of blastomeres are formed. I hope you got the idea about how cleavage take place in the eggshell. The outcome of the cleavage is termed as cytoplasmic localization, and it seals the developmental fate of cell descendant. Means the cell that is later on, what organ will be or what term will be formed, it is localized and at that stage. In the next lecture, we will study about the later event of the development of chick, that is morula, blastula, gestulation and neuralization.